April 11th, 2019. A spacecraft is about to land on the moon. The engine fires. The descent begins. Then the engine fails. Beresheet slams into the lunar surface at full speed. The mission is a total loss. Israel's first moon mission, years of work, gone in seconds. But here's what nobody in mission control knew. Buried inside that spacecraft, secretly, without official approval, were thousands of living creatures. And they survived. This is a tardigrade, a water bear, the toughest animal ever discovered on Earth. It's smaller than a grain of sand. You've probably walked past millions of them without knowing they existed. But this tiny creature has been hiding a secret for 250 years. And that secret, discovered just 9 years ago, might protect cancer patients, might let humans survive a trip to Mars, might one day change your life. This is the hidden story of the water bear. The year is 1773, Quedlinburg, Germany. A pastor named Johann August Ephraim Goes has a problem. He is obsessed with the invisible world. He sold part of his own library, his most precious possessions, to buy a microscope. And every evening, instead of reading theology, he is peering into pond water. One night, in a clump of wet moss from behind his church, he sees something no one has ever described before. A tiny 8-legged creature, waddling, like a miniature bear swimming through water. He names it Kleiner Wasserbear, the little water bear. He publishes his observation. The world shrugs. It's a curiosity, a strange little animal in moss, nothing more. 3 years later, an Italian polymath and Catholic priest named Lazzaro Spallanzani, the same man who proved bats navigate by sound, gives it a proper scientific name: tardigrade, slow walkers. Neither man had any idea what they had found. They planted a fuse, and it would burn for 250 years. Now, before we go any further, you've heard about tardigrades. The internet has a story about them, a story that is almost completely wrong. The story goes, tardigrades are immortal. They can survive anything. They are indestructible. And here's the twist. In their active state, when they're wet, when they're alive and moving and looking for food, they are fragile. At normal mammalian body temperature, 37 degrees Celsius, the temperature of a fever, a wet, active tardigrade dies within 48 hours. You could kill one with common insecticide, with a sharp instrument, with a warm shower. The creature we call indestructible can be killed by a warm day. So how did they survive the moon? The answer is a state called cryptobiosis, and inside it is one of the most extraordinary biological tricks ever discovered. When conditions get bad, drought, freezing temperatures, radiation, the tardigrade does something no animal should be able to do. It expels 99% of the water from its body, contracts into a sealed barrel, pulls its legs inside, and waits. In this state, the ton, its metabolism drops to 1/100% of normal. All detectable biochemical activity stops. To every measurement we have, it appears dead. But inside those cells, something extraordinary is happening. Special proteins, called Koch proteins, begin to polymerize. They form filaments throughout the cytoplasm, and those filaments solidify the cell interior. Not ice, not gel. Glass. The tardigrade vitrifies its own cells. The same process that preserves insects in amber. The cell is not destroyed. It is suspended, frozen in time without ice. No ice crystals tearing through membranes. No chemical reactions degrading proteins. No clock ticking. In this state, a tardigrade is not alive, but it is also not dead. It is waiting. And those tardigrades on the moon, they're still waiting. Right now, in the debris field of the Beresheet crash, no water to rehydrate them. No signal that Earth still exists. Waiting. You might be thinking, survival on the moon is impressive. But it's passive. They just go dormant. How hard can it be? In 2007, a Swedish professor named Ingemar Jonsson decided to find out exactly how hard it can be. He convinced the European Space Agency to strap a container of 3,000 tardigrades to the outside of a satellite and launch it into open space. No capsule, no shielding, no atmosphere. Directly exposed to hard vacuum, extreme temperature swings, cosmic radiation for 10 days. When the container came back, Jonsson opened it. Most of the tardigrades were alive and reproducing. His words, the space vacuum, which entails extreme dehydration and cosmic radiation, were not a problem for water bears. It was the first time any animal had survived direct exposure to the combined vacuum and radiation of open space. But here's what stopped him. The ones exposed to full-spectrum ultraviolet radiation, the unfiltered UV of direct sunlight. Those died. The toughest animal on Earth has a weakness. Unfiltered solar UV will kill them. And that's the thing about water bears. Every time you think you understand them, they reveal a new layer. September 21st, 2016, the University of Tokyo, a researcher named Takuma Hashimoto publishes a paper that changes everything. For years, scientists assumed tardigrades survived radiation by repairing DNA damage, fixing breaks after they happened, like a repair crew patching roads after an earthquake. Hashimoto's team sequenced the full genome of Rhamazadius varionatus and found something that didn't fit that model. A protein, a single protein with 445 amino acids, largely disordered, no fixed three-dimensional shape, and when they searched every protein database in the world for something similar, nothing, not in bacteria, not in plants, not in any animal, not anywhere in the tree of life. They named it Dsup, damage suppressor. And here's what makes it extraordinary. Dsup doesn't repair DNA damage. It doesn't neutralize radiation. It doesn't do anything active at all. It simply wraps itself around the DNA double helix and sits there, using electrostatic attraction to coat the genetic code like a physical shield, blocking hydroxyl radicals and X-rays before they can reach the DNA. Passive protection, more effective than any active defense biologists had imagined. Then Hashimoto's team did something daring. They took this single tardigrade gene, and they inserted it into human cells. The human cells with Dsup showed 50% less X-ray damage than the cells without it. A protein from a microscopic pond animal, one that shouldn't exist by any known biological logic, was protecting human DNA. We need to find the molecules that allow tardigrades to tolerate such conditions, Kunida said. They found one, and they had no idea where it would lead. In the United States, more than 60% of cancer patients receive radiation therapy. Radiation is powerful, but it doesn't discriminate. It damages tumor cells and healthy tissue at the same time. The dose doctors can give is limited by how much healthy tissue can tolerate. Many patients never fully recover from the damage. Some must stop treatment early. The tumor survives because the body can't handle more radiation. At February 26, 2025, a paper from MIT and the University of Iowa, Giovanni Traverso and James Byrne delivered DNA damage. It doesn't neutralize radiation. It doesn't do anything active at all. It simply wraps itself around the DNA double helix and sits there, using electrostatic attraction to coat the genetic code like a physical shield, blocking hydroxyl radicals and X-rays before they can reach the DNA. Passive protection. more effective than any active defense biologists had imagined. Then Hashimoto's team did something daring. They took this single tardigrade gene and they inserted it into human cells. The human cells with Dsup showed 50% reduction in radiation-induced DNA damage, without reducing the effect on tumor cells. The water bear protein protected the healthy tissue while letting the radiation do its work on the cancer. Radiation can be very helpful for many tumors, Traverso said, but we also recognize that side effects can be limiting. Human trials have not yet begun. This is early. Here's the thing about Go's little water bear. He found it in 1773. He described it carefully. He published his notes, and then he moved on. He never imagined what it was. No one did. For 2 centuries, tardigrades were a curiosity, something biologists cataloged, something unusual, something you mentioned if you wanted to seem interesting at a dinner party. Then, slowly across different continents, different decades, different questions, the chain began. The space biologist who wanted to know what could survive open space. The genome detective who found a protein with no relatives. The cancer doctor who asked, what if we could protect patients from their own cure? None of them started from the same question. But every answer led to the same place. A creature smaller than a grain of sand, turning its cells to glass to wait out the end of the world, shielding its DNA with a protein evolution, spent 500 million years designing. We spent 250 years marveling at it. We are only now beginning to steal its tricks. Next time, you see a clump of moss on a roof, a wall, a park bench, know this. There are water bears in there right now. Waiting, carrying secrets we haven't found yet. Speaking of biology doing the impossible, there is a trial, one of the most replicated findings in all of medicine, where doctors gave patients a completely fake pill. No active ingredient, no drug, nothing. And the patients got better. Not because they were fooled, not because the pain was imaginary, because something inside them responded. Something real. Something that has baffled neuroscientists for decades. The same body that hides radiation shields in a pond animal can apparently heal itself with a lie it tells itself. The placebo effect is not a curiosity. It is one of the most powerful and least understood forces in medicine. And the story of how we discovered that and what it means for you is